This morning, I would like to introduce Ken Burns, who will be introducing our speaker for today. Thank you.
Good morning. I'm filling in this morning for Pushpa. I've had a major personal problem during the past week, and so it's my pleasure to introduce today's speaker, Professor Chris McCurdy, who's in the Department of Medicinal Chemistry and Pharmaceutics, and is director of the UF Translational Drug Development Core. Dr. McCurdy received his Ph.D. in Medicinal Chemistry from the University of Georgia and completed a fellowship in the Department of Medicinal Chemistry at the University of Minnesota College of Pharmacy. His research has focused primarily on drugs or compounds to treat pain and drug abuse. And so especially with respect to the opioids. And he's an internationally recognized expert on Kratom, K-R-A-T-O-M. I would not pronounce that from him this morning. And that's a nice compound that's extracted from the leaves of a tropical tree. I just want to add that in addition to his background that I described, he has served as president of the American Association of Pharmaceutical Scientists and also serves as a consultant to the U.S. Food and Drug Administration's Drug Safety and Risk Management Advisory Committee. So I think at this point I will turn the mic over to Chris and let him give you some real facts about the subject. Chris, welcome.
Sure. Thank you very much, Dr. Burns. Really a pleasure to be able to join you all today. I really wished I could have been there in person, but I also want to be respectful and cognizant of how things have been going here in terms of the COVID and the uptick in cases around the country, but particularly in our own backyard. And so I really would have preferred to be there talking with you all in person and enjoying some time together. But this is the way that the world has sort of become, and we've learned how to live through Zoom. And to be quite honest, it's been a good educational tool for us, and it's been a way to travel around the world without traveling, unfortunately or fortunately, however you want to look at it. So I'm happy to be here today to talk a little bit about Kratom. It's pronounced several different ways. You may hear it in the news or in the label as Kratom, and I look at it as tomatoes and tomatoes. And so it's Kratom. The way it's pronounced in Thailand, where it's originating from, is Kratom. So they actually put sort of a emphasis on the tom part. But we'll talk about what Kratom is as we move through this. The picture on the screen that you can see right now is actually a stand of trees in Malaysia that I was actually able to visit and photograph a plantation that's there and in use for individuals around the upper part of Penang, Malaysia. So we'll talk a little bit more about the tree in just a moment. I am a medicinal chemist, and so I have a big interest in chemical structures. And we'll talk a little bit of chemistry today. I promise I won't overdo it, but we'll definitely look at a little bit of chemistry as we move through. The compound in this the bottom right corner of the screen is called mitrogynin, or some people will call it mitrogynin. And it is the major compound that's been isolated out of this plant material, and one that's received a lot of the attention in terms of research, mainly because it's the most abundant. And usually in natural products, when a compound is most abundant in the plant, it's then assumed to be the primary active constituent in that plant. And so I'll tell you a little bit about what we've learned along the way in terms of it being maybe of interest or maybe not of interest as we've developed into this. So just to give you a little background on how I got into researching this plant, my expertise was really formed around opioid chemistry, particularly that around morphine and the compounds that are isolated from the opium poppies. That got me interested in many compounds that could interact with opioid receptors. And over the years of my career, I had worked on other plants that have opioid activity. But this plant, Kratom, really excited me. For some reason, it had received a lot of attention, specifically by SmithKline and French, which is now GlaxoSmithKline Pharmaceuticals, in the 60s. And they did the most complete and thorough studies, I still believe, on this molecule, the main alkaloid that exists in the literature still today. And what they determined was that the pain-killing ability of this molecule was basically similar to codeine or similar to aspirin. And at the late 60s, early 70s, they didn't see a potential in the marketplace to really pursue this as a profitable product. And so they gave it up and sort of abandoned development of it, even though it appeared to be a fairly safe and effective molecule. So I saw that and was really curious as to why nothing was being done with it. And so we started digging into what's going on with this product. And what we know is that it's in the name of Mitrogyna speciosa. So that's the formal taxonomy name. It is in the family of the Rubiaceae. Rubiaceae family also includes the coffee plant. And we'll talk about some of the similarities, but very much dissimilarities between Mitrogyna species and our typical coffee drink. And again, this genus and species, Mitrogyna and speciosa, there's about 7 different species of Mitrogyna, but supposedly, according to the literature, Mitrogyna speciosa is the only one that contains mitrogynin, that active alkaloid constituent I referred to on the previous slide. So this is a tree that seems to be indigenous to Southeast Asia, particularly Thailand and Malaysia. And you can see the red oval in the middle of the map there on the screen, which is showing you the border between peninsular Malaysia and Thailand. And that's where we believe is sort of ground zero for where this tree originated. And that's where we see most of the use in Southeast Asia, and traditional medicine resides there in the history. And I'll speak a little bit about that traditional use here in a moment, because there's a big difference between how the plant is used traditionally and how it's being utilized in the Western world, in Europe and in the United States. And that's really been a focus of a lot of the work that we've been attempting to do. So again, it's referred to as Kratom or Kratom in Thailand. It's called Biak Biak or Kitom in Malaysia. So Kratom or Kitom, just depending on which side of the border you're on, but they all basically understand that same language. Up until this point, there's been about 40 reported alkaloids. So not just that major alkaloid I showed you, mitrogynin, but another 39 that have been reported to date. We really want to sort of revise that and revisit that because we believe that there's not that many distinct alkaloids within the plant. But when you do natural products isolation and you put these plant materials through lots of chemical acid and base changes to extract these molecules, there's chemistries that can happen, particularly in different solvents like alcohols, that can cause modifications of the molecules, the naturally occurring molecules, and you can isolate something that appears to be novel and from the plant, but really is not from the plant. So I would say there's probably in the neighborhood of 20 to 25 alkaloids that the plant does produce. We're working with a group at the Max Planck Institute in Germany to do the biosynthetic pathway and discover all the genes and proteins that are involved in the synthesis of these alkaloids with them. And we're learning that many of the reported compounds are really not existing in the plant machinery. So we can talk about that more in the question and answers if somebody's got an interest. The genus Mitrogynin was actually named for the shape of the leaf, and you can see the leaf at the top there. It's shaped like a mitre hat or a bishop's hat if you sort of rotate it backwards 90 degrees. And that's pretty typical of the Mitrogynin. The leaf shape all looks like this, sort of long and then pointed at the tip. The tree flowers. It is a self-propagating tree, we believe. We're doing a lot of studies, and I'll talk about that as well, in IFAS here at UF to really understand this tree and how it grows and how it can be propagated. There's a lot of commercial potential in this plant as well. So we'll talk about that a little bit more as we move forward. I just really want to set the scene in the background as to how everything is understood, and then we can talk a little bit more in depth about the science. Kratom or kratom and mitrogynin, that major alkaloid, is really a tea that is prepared in the rural parts of that border region of Thailand and Malaysia. And it's used by outdoor laborers or field workers to really relieve their pain, also to help stimulate them, to improve their work capacity, to improve their ability to work within that hot, humid, equatorial type tropical environment. But interestingly, it's also been used to reduce opioid withdrawal, or actually as a substitute for opium. So many of these individuals may use opium for pain relief, and they've actually, when they run out of their opium, they would switch over to this kratom tea and utilize it, go into withdrawals until they could get opium again. Later, it was learned that they could switch over to this tea and get rid of using opium altogether and have a much better life quality, where they would have this sort of mood elevating properties, the stimulant properties that are associated with kratom. But in addition, they got the pain relief that they were seeking from opium previously. And so it's a very interesting product. And there's a picture of another tree down in the left that's flowering. Up in the right-hand corner, the traditional preparation is shown. And what they do is every morning they will pick up to 1 kilogram of leaf material off of the tree, so fresh wet leaf material, and they'll put that into 5 liters of water. And that kilogram of leaf material is boiled for anywhere from 2 to 3 hours. Essentially, when you get down to about 1 to 1.5 liters of what they call juice, they don't call it tea, juice left. And then they package that, they let it cool, and then they package that into bags. And these bags are what are purchased by the laborers. Now, interestingly, it's illegal to sell kratom in Malaysia. So there are roadside stands that sell it. And most of the time the police leaves them alone. But every now and then the police will make an example of one of the roadside stands and charge them a fine. However, the fines are so negligible that they can pretty much make back the fine cost within a couple bags sales. So that really is not a deterrent to them producing this tea. So the tea, you can see as it boils, it's a nice clear solution. As it cools down, you get some precipitation of some of the compounds that are in that aqueous solution, that water solution, that tea. And then that bag, or sometimes they'll package it in plastic bottles, water bottles as well. But that bag serves as a daily dose, if you will. They will take that bag and divide it into 3 doses throughout the day. Sometimes just 2 doses, sometimes 4 doses. It really depends on their need. They just titrate the amount they need for the effect they're going for. And again, they're going for this effect that's really more of a stimulant, mild pain relief effect to carry out their daily activities. So they really look at this as a beverage like coffee, and they use it in that capacity. It's a social beverage there as well. So I mentioned these sort of roadside stands. I should have put a picture of one of the roadside bars in here for you to see in Malaysia. But we stopped at one of the roadside stands and talked to some of the users that were there, and really it's just going on like a normal coffee shop. We do think there's really great potential, and I'll try and show you some of that for using Kratom in reducing opioid use, or getting people off of prescription opioids or illicit opioids. And I'll convince you, I hope, that Kratom itself is not an opioid, even though the FDA is coming out and saying that it is. I've been invited to speak to the FDA several times on this topic as we learn more and more information about the science. And the FDA just 2 weeks ago released a contract request for the first time to do some human clinical studies with Kratom. And so it's very interesting to see that the FDA is interested now in pursuing maybe some potential here. Obviously, the National Institute on Drug Abuse has been very interested. They've funded us as well as other laboratories to study this plant and really understand what the potential is. And so we'll continue talking a little more about that as we move through. Sorry. What was more interesting for us, at least, was in 2020, we published a paper in collaboration with our collaborators at the University of Science Malaysia, showing that polydrug users, so individuals that are using opium or prescription opioids along with methamphetamine, were actually utilizing Kratom to reduce use of opium and methamphetamine. And so this was encouraging because there's absolutely no FDA approved treatments for methamphetamine. And if we can reduce use of methamphetamine with this product, it might be another interesting application. So we're in the process of trying to study that as well and understand if there really is a role in methamphetamine reduction this plant. Again, we already talked about the predominant active ingredient is mitrogynin. This is, if you want to get technical into the chemistry of it, it's an indole-based alkaloid. This is an indole aromatic ring system. If we extend that a little bit farther, this is called a coinanthine ring system. And then one consistent thing that we'll see as you start to see the rest of the chemistry, and I like to play with my kids, which one is similar, which one is dissimilar, because we'll see it as we start to see the other individual alkaloids that we've isolated and work with. There are parts of the molecules that are very similar, and there are parts that are very different. So we'll point that out. And I have some thoughts on why that might be the case, as to why the plant is producing so many molecules with so many similar chemical properties. So that's the use of kratom in the traditional sense. Kratom use in the USA has gotten a very negative read in the news and in a lot of the medical literature. The medical literature has reported bad case results. So individuals that end up in an emergency room or a poison control center with a bad experience from utilizing kratom are the ones that end up in the peer-reviewed medical literature as case reports and anecdotal studies. We have been working really hard, as have other groups across the United States, to really investigate the pharmacology, the toxicology, the pharmacokinetics, and what all is happening with these products. And also why we see harm in the United States, but we don't see harm in Southeast Asia where it's traditionally used. So part of the original question for me was at least just look at what's available in the United States. So this is a picture of various products that are for sale in the United States. Is widely available across the internet, smoke shops and vape shops. You can go down on University Avenue in Gainesville and go to any of the smoke or vape shops, and they'll have kratom products available for sale. In June of 2019, which is the latest data we have, the American Kratom Association, which is a sort of advocacy group for kratom itself, but also is really trying to act as a regulatory body for assuring that kratom products are clean, unadulterated, and true to the labeling in the marketplace. But they unfortunately can't regulate the marketplace. And so the marketplace is full of things that are not kratom, but are labeled to be kratom. But regardless, they have reported almost 2,000 metric tons are exported to the United States every month. And that was 2 years ago. We know that opioid use and drug use has increased during COVID-19 lockdown. Opioid overdose deaths have gone up 30% just in the last year. And so there's a great need to really understand if this has some potential to really help in the opioid crisis that we're facing. But 2,000 metric tons exported to the US every month is indicative of a large population of use. And what we've determined, along with the American Kratom Association, we've gone and checked their math. The typical dose that individuals will ingest is 3 to 5 grams of leaf material, which is usually reduced down to a fine powder. And they'll make teas out of that themselves. And if you look at a typical dose of 3 to 5 grams, and then you do the back calculations at 2,000 metric tons, there's somewhere in the neighborhood of 15 million or more users of kratom in the United States. So it becomes a pretty significant issue. And if you haven't even heard of it yet, that's not a surprise to me either. But it's starting to become more and more popular, particularly amongst those individuals that are really trying to get off of prescription opiates or illicit opiates. And there's a great percentage of the population of users that are using it in that legitimate fashion and are having no issues with use. However, there's the small group of users that end up trying to get a legal high from this, since it has been called out as an opiate. And there's a lot of abuse and there's a lot of over ingestion. And I mentioned that the traditional users in Thailand and Malaysia will titrate themselves. And so they'll drink enough of this to get the effect that they want. If they drink too much of the tea, they say they get nauseous, and that's when they know that they're getting to a point where it's too much for them. So many of them don't have that issue in terms of overuse in Southeast Asia because it's been part of the culture for so long. But here, unfortunately, you can see there's products that are targeted that look bad, right? For you. I mean, that's a name that sounds like it should give you a euphoric feel, good result. Some of these, it may not be easy to see in terms of their names, but they've got different ones called Black Panther and Viva Zen. Viva Zen was actually originally labeled as Vicodin, so it sounded like Vicodin. And the FDA did come out and make them change their labeling, at least for that. And so there's this real confusion with the FDA. The FDA has enacted import bans on this material. And so any material that's imported into the USA is illegally imported into the USA. But you see here, there's 2,000 metric tons coming in every month. Most of it is coming in under the guise that it is sodium alginate, which is essentially seaweed or a thickening agent that's used in cooking. And it's really impossible if you look at ground kratom leaves that are green and you look at sodium alginate powder, they look very, very similar. So it's no surprise that they're trying to get around the system. And quite honestly, the FDA doesn't have enough staff to be out investing in their time in monitoring all these ports. So there's a lot of material coming in. That material is coming across a long way from halfway around the world. And it's being exposed to, different environments as it's shipped across the globe. And so there's heat factors that come into play. There's humidity factors that come into play. There was an outbreak of salmonella in kratom products a few years ago. That the FDA, again, warned people from using these products because they're not tested very well and they're not regulated. But as I mentioned, the American Kratom Association has been doing a large effort to try to make a very similar product available in the United States to what's used traditionally. I think that's pretty difficult. It's not impossible, but the issue is that these powders, these dry powders, are very, very different than the fresh leaf material product that you see traditionally used. Not only that, there's been concentrates made and extracts made, and so they've tried to increase the amount of alkaloids in products that are available in the United States and in Europe, which obviously pushes problems as you ingest more concentrated amounts. You're exposing a body to more material, and so this can be, of course, a very negative situation, and has resulted in some negative situations. Because of that, and I just now realized that I didn't have the slide in this slide deck, I will just tell you what we have done. We have been able to obtain, and as we, I mean the University of Florida, has been able to obtain germplasm for these trees, and we are growing trees in Apopka to really study the difference between natural grown, freshly prepared ethnomedicine, if you will, versus what's available in the marketplace here in the US, and really dig into differences in chemistry and differences in use. But there's also a great interest in the state of Florida looking at alternative crops right now, particularly with the greening virus that's present in the citrus industry, and we're losing citrus orchards pretty readily. There's interest in industrial hemp, and the University of Florida had an industrial hemp program. I don't know if you've had anybody speak to you about that. Our laboratory was involved in that program as well, and doing chemical analysis for that as they were doing the test fields. And we're doing the same thing in terms of trying to understand how mitrogynin speciosa is grown, the tree, how it can be propagated, how it can be harvested, and what commercially there could be from this if we can derive a botanically based medicine. So there's a lot of work going on, and there's a lot of hope that we may be able to come up with a product that can be applicable to the opioid crisis. I don't think it will be the end all be all, but it may be something that can help. And now I want to get into a little bit more of the science and try to convince you that it's not entirely opioid based, although it'll start out that way. But there's a lot of other neurotransmitters and a lot of other chemical systems that are being engaged in the body by the alkaloids from this plant than just opioids. So we'll move on here to show you kratom and mitrogynin again. What are the things that we're looking at in terms of cases? And I mentioned there's been some negativity around this, but very few deaths, if really any, are solely attributable to kratom alone. We do know that certain materials that are available in the marketplace have been adulterated. We have had individuals show up at emergency rooms around the country, and we've been able to get the samples of material that they were ingesting, and we've seen kratom products laced with morphine and laced with hydrocodone. I just got an email this week, and we're in the process of getting materials to us about fentanyl being laced in a product. And so it's quite frightening. It's scary. There's really no regulation around the botanical supplement or dietary supplement industry. And this is just another example that even though there are many vendors trying to do the right things and do things on the up and up, there are some and individuals out there that are nefarious and doing business to make a quick buck at the expense of whatever that is. And that's the sad part of being in this area. But very few deaths can be attributed alone. Most all of the deaths that have been reported in combination with other substances, particularly drugs of abuse, and that could be a contributing factor to those case reports. So mitrogynin itself, we can hear about this alkaloid. The major alkaloid in the plant is actually a partial mu-opioid agonist. And so what this means is something like fentanyl or oxycodone or morphine is a full opioid agonist. In other words, I like to explain this when I'm teaching my students. A full agonist essentially turns a faucet on full blast. And so you're getting all the chemical signaling into the cellular pathways and into the neurons that you could possibly have. A partial agonist, obviously, is just partially opening up that valve. And so you're not getting the full onward effect. And so it actually only stimulates about 40% of the maximal effect. And this is getting a little bit technical, but what mitrogynin is being touted in the literature is a biased agonist. And so opioid receptors are G protein coupled receptors. In other words, when they are activated, there's a protein called the G protein. It's a trimeric protein. So it's 3 proteins that come together. And when that receptor is activated by a compound, say like morphine, that causes a conformational change to the protein that allows for the G protein to be activated, dissociate, and then carry out downward streaming signaling in the cell that then perpetuates the effects that you see. So there's another protein called beta-arrestin, which actually can come in and arrest that signaling pathway and cause internalization of the protein from the membrane and stop what's happening. That is thought to be involved with many of the side effects. So this beta-arrestin recruitment is thought to be involved with many of the side effects that we see from prescription opioids in terms of respiratory depression, constipation, and even potentially abuse liability. So the whole interest that mitrogynin does not recruit this beta-arrestin protein, there's a lot of interest in the pharmaceutical industry because these molecules are being targeted in development as opioids with low respiratory depression and constipative effects. And indeed, kratom and mitrogynin itself do not seem to have very severe respiratory depressive effects associated with them, which is very different from your typical opioid pattern. And so this work was not originally published by our lab at Florida, but it was published by some of our competitors that are at Columbia University, Andrew Krugel and Davor Sims. Good friends and good competition, and we keep each other on our toes, that's for sure. So therapeutic potential. Where are we trying to go with this? And as I mentioned, we're hoping that this could serve as a potential treatment in opioid detoxification. It could replace several medications that are used currently for opioid detoxification. And that's because it has very distinct pharmacological actions, not just opioid, but it has adrenergic actions. And I'll show you some of that precise data here in a moment. But if you were able to use a capsule of kratom or a cup of tea of kratom instead of ingesting 3 or 4 different medications, there's a thought that it can improve medication adherence and really completing the detoxification process. So that's different. This is going through detox and withdrawal. We do know that there is a lack of opioid-like overdoses in humans. And again, this we think is due to the mechanisms of action of many of these molecules, and I'll talk about that more in a moment. Most of the overdoses that are called into poison control centers actually resemble stimulant overdoses. So cocaine-like or methamphetamine-like overdoses versus an opioid-like overdose. So what does that mean? It means that individuals are having seizures or tremors or overheated, sweating excessively, instead of having respiratory depressive type effects, which is what you would get with an opioid overdose. So very strange reports that people didn't really understand coming out of the poison control centers. We also think that there's medication-assisted therapy place for kratom to be used to reduce opioid use overall. Kratom withdrawal, it can be addictive itself. There haven't been any very good clinical studies on that, but we do know that there are individuals in treatment facilities being treated for addiction to kratom, just like there are individuals in treatment facilities being treated with kratom for addiction to opiates. And so it's a very interesting, paradoxical thing that we're seeing. I think that it's mainly based on the quality of, I'll touch on that here in a little bit as well. But kratom withdrawal has been reported to be very mild. So SALS is the subjective opiate withdrawal scale. It goes from 0 to 30, and anything below 10, and it's essentially you self-identify symptoms that you're having with numbers, and then those are added up and you get an overall score. So the average score is actually less than 9 on this scale, which is somewhat comparable to where coffee falls, is my understanding, versus something like prescription opiate withdrawal, which would be closer to 30 on this scale. So there's mild, moderate, and severe withdrawal. Mild is anything under 10, moderate is 11 to 20, and then severe is 21 to 30 points. And I don't have an example of that questionnaire for you, but it probably can be found easily online if you want to Google that later. And again, polydrug users seem to report that kratom may reduce methamphetamine or stimulant use. Is here really come down to the fact that no standardized product has presented itself in the marketplace, and there's no rigorous clinical trials to evaluate these claims. So a lot of the work that we're trying to do is really develop a standardized formulation and be able to go into human clinical trials and really examine some of these potential therapeutic uses. The picture, by the way, behind me in the background is one of the greenhouses down in Apopka, and those are all mitrogynin speciosa trees that are growing. And those particular trees from the greenhouse are in the field now, and we have about 80 trees in the field and another several hundred still in production in greenhouses. So we're really trying to understand that. So now I'm going to dig a little bit more into chemistry, and then we'll move into pharmacology and a little bit of pharmacokinetics. And I'll talk about what we think is actually going on and what's happening when someone ingests this product. And I want to stress that Kratom, as you saw it already, is a natural product that contains several compounds. So we're focused on alkaloids here, and alkaloids just mean that it's a nitrogen containing compound. We're focused on those for now, because usually those are the things that are biologically active at CNS drug targets. If we go back through natural product medicine, that's been historically the case. One glaring example that contrasts that is the cannabis, where none of those cannabinoids contain nitrogens. So they are very obviously CNS active. Particularly THC, but we're not focused on any of the other types of compounds at this point, just because traditionally alkaloids have been the primary mode of action. And so these are compounds that we've actually isolated, characterized, identified, and studied in our laboratory. Again, mitrogynin being the major alkaloid. And this is where I was talking about comparing the Sesame Street, sort of which one looks like the others. But what you'll notice, I hope quickly, is that every single one of these molecules has this same functionality on the bottom right side of it. And that's what we call a beta-methoxyacrylate moiety. That portion of the molecule is retained in every single alkaloid that we've seen. It could be cyclized in a couple of them, but for the most part, we see it in every compound. And so that leads you to think that it's there for a reason, for the plant's protection, and something is going on. And we're pursuing that now in IFAS with other researchers, trying to see if this is fungicidal, herbicidal, insecticidal. What purpose are these types of molecules serving the plant with? Because the plant doesn't care if it's producing opioid or serotonin or mood enhancing chemicals. It's producing those chemicals for its own purpose somehow. So just to tell you, we've isolated many of these compounds and studied them, and some of them are very much identical. And so if we just take a look at mitrogynin and spigenin, for example, these molecules are identical in every aspect except for one stereochemical position. So this portion of the molecule is the left hand and the right hand version, or mirror image sort of each other. And so we also noticed that these have drastically different pharmacologies. But in the original isolation of these molecules and the original quantification of these molecules, because these have the exact same molecular weight, many times mitrogynin content has been over quantified because you have speciogynin. And if we look to the right of that, speciosialatin, which is again exactly the same molecular weight, but now differs in the stereochemistry around this carbon atom, where the hydrogen is coming out of the screen instead of back away in the screen. And I won't go into any more details of chemistry. If somebody's really interested, we can have that discussion later, but most of the time it bores people to death, so I do that to all of you. So let's just move along. But I want to walk through some of these that we've tested and show you that they're not just opioid molecules. And so what we've done is we've contracted with a company called Eurofins that will screen 82 different common drug targets in the central nervous system. And what they do is they take a high concentration, so 10 micromolar concentration, and they do a preliminary screen to see if there's any interaction with these drug targets. And then if they do get a hit, which you can see here as defined by the X's, and what a hit means is that they displace 50% or greater of a radioactive material that's selective for that drug target off of that drug target. And so that means that that compound is coming in and competitively displacing the ligand that they're using to measure for affinity of that target. So we're seeing these molecules interact with these proteins. And the proteins that we see being interacted with are 5-HT or 5-hydroxytryptamine serotonin receptors. So we see a couple of serotonin receptor subtypes. We see alpha adrenergic and beta adrenergic and dopamine receptors. And these are all involved in either dopamine signaling or your adrenaline or epinephrine type activities. So stimulant type work. And then we see kappa and mu opioid. So there's 3 different opioid receptors. Mu opioid receptor is the main target for the clinically used opiates. And then we see a couple of other ion channel targets here. I won't go into any of these in detail. But what happens is you test these at this high concentration, 10 micromolar or 10,000 nanomolar concentration, which is very high, more than likely not going to be achievable in a dose that's used. But we look for that for a hit, and then we go down to 100 nanomolar, so much lower concentration, to see if we can still displace that radioactive molecule. And here you can see that at that dose, nothing gets done. So why is everybody so excited about mitrogynin and claiming it's the most active compound? Because under the screening system, it doesn't really show much. Well, we took that information and actually calculated out affinity labels, what we call Ki values. This is showing you the constant that we can create. So the concentration of the molecule that actually is interacting with that target. And so we do see that it does interact with mu opioid receptors. It interacts with kappa opioid receptors. It does not interact with delta opioid receptors very well. It interacts with adrenergic receptors. And again, adrenergic receptors are the site for many of the stimulant effects that we can see. These are also used clinically. These are targeted for treatment of hypertension and other areas. Interestingly enough, some of the anecdotal reports of users in the traditional sense say that kratom regulates their blood pressure. It also regulates their sugar. And so we've gone into looking in some models of diabetes. I won't go into that today, but there's some interesting data coming out around possibilities of helping to regulate blood sugar by affecting the microbiome in the gut a little bit with these compounds. And so again, there's just so many layers to this based on the anecdotal reports that we see. But we do see that these molecules are activating alpha receptors or blocking alpha receptors alpha-2 receptors are specific targets for drugs called clonidine and lofexidine. Lofexidine was the most recently approved drug by the FDA to treat opioid withdrawal. And so this is really interesting for us in that we're seeing some of the pharmacologies play out in terms of the way that the treatment, the clinical treatment is playing out as well. Again, we use mu opioid partial agonists to treat opium addiction or opiate addiction. Buprenorphine is one of those compounds, and methadone is another one. They don't produce the euphoric effect that you get from something like morphine or heroin or fentanyl, but they are partial agonists. They activate that receptor just a little bit, like turning on the faucet partially, right? So there's a lot of pharmacology we could dive into and dig into, but this was interesting to us because, as I mentioned before, poison control centers say that kratom overdoses resemble stimulant overdose and not opioid overdose. And we see that that could be a result of some of these interactions. Another one of the compounds that is reported to be in the plant is called 7-hydroxymitrogynin. And so the only difference between this compound and the compound I was talking about previously is this OH or hydroxy group that's been inserted into the 7 position, and that's why it's 7-hydroxymitrogynin. This compound is reported to be present in about 1 or 2% of the leaf material when it's been isolated from leaf material. We have never seen it in fresh leaf material. We cannot find an enzyme that produces it in the plant in the work that we're doing with the Institute in Germany. We believe that this molecule is generated as a post-harvest artifact. So after the leaves are picked and the leaves are dried, there's an oxidative process that happens to those leaves, and this oxygen gets inserted. Now, the crazy thing is, this turns mitrogynin into an incredibly high affinity and very potent mu opioid agonist. So right along the lines of something like oxycodone, OxyContin, or fentanyl. So it's a full agonist. It's very concerning that it can be there. And this is what I'm talking about in terms of the quality of products that are coming into the United States. So we're seeing materials that are coming in that have no detectable levels of this 7-hydroxy product. And then we're seeing materials come in that have huge amounts of this 7-hydroxymitrogynin content. And that, in my mind, is probably what's having the issues in the marketplace, where individuals are getting addicted to this product because there's a highly addictive substance inside that product that's in a greater concentration than is in many of the products that are in the marketplace. And I'll show you some data around the addictive potential of this as well. So I also want to emphasize again that this plant has all these compounds in it at once. And so I think of it as a symphony orchestra, and you've got all these different instruments or molecules playing out their pharmacological opus, if you will, at the same time. And they're in various concentrations. So it's like having a huge string section in an orchestra, maybe mitrogynin. It's not as powerful as the brass section that can just blare out with minimal instruments. And so those concentrations of each of the molecules becomes important in terms of their pharmacology and how then they blend back together and play together. So we take 7-hydroxymitrogynin out, purified it, and we can listen to it full blast. We know it's an extremely potent, highly addictive opioid. But in the context of the entire plant material, the amount that's there, unless it's been elevated to extremes, usually is of consequential fact, and there's nothing really to be concerned about. So I'm going to try and go through this detailed pharmacology a little bit quicker, just to show you what's happening. There's been reports that mitrogynin, the major alkaloid, is more potent when it's given orally than if it's given subcutaneously or intraperitoneally. So if it's injected into the animal, it's less potent than if it's given by oral route. And orally is how kratom is taken, as we can see. And so this compares morphine in blue circles, and what we're looking at is dose across the bottom, so increasing doses to the right, and the percent antinociception, so how much pain killing property it has going up the left-hand side axis here. So what you see is morphine, as you increase the dose, you get more analgesia, more pain relief. 7-hydroxymitrogynin, which I just told you was incredibly more potent than morphine. It is. It takes much less of 7-hydroxy to give you pain relief. And then mitrogynin is pretty not nearly as potent as those two molecules. However, if you give mitrogynin orally, what you see is a change in that dose response curve, and it becomes more potent. It becomes almost as efficacious as morphine. So it's crazy, right? What's going on? Why would be ingesting it through the oral route versus directly injecting it into the body cause some difference? And then we tested kratom, that full symphony orchestra orally, and you can see it can reach full analgesic potential, but it takes quite a bit more of it to do so. So what's going on here? Well, the immediate thing that you have to think of is if we're taking it orally, all drugs that are delivered orally are subject to what's called the first pass effect. And that first pass is going to the liver first. And the liver is going to metabolize that material. And so the liver is metabolizing mitrogynin into something potentially more potent as it's delivered orally. So we wanted to look at that. And so what we did was we took rat liver microsomes. So we isolated metabolizing enzymes from rat liver. And we also looked at rat intestinal microsomes because the intestine has the ability to also start metabolizing before we absorb that material into the blood supply. And what you can see is we dosed these, really they're test tube studies. We put mitrogynin into that test tube and we look for the formation of 7-hydroxymitrogynin. And so now we know that 7-hydroxymitrogynin, this problematic potential opioid, is actually being generated in our own bodies through the liver. And it's being generated in the intestine. And maybe that's why it's much more potent in terms of its analgesic activity when it's given orally. So percentages converted, we did a lot of fancy pharmacokinetics to really determine that about 9% of the dose, oral dose of mitrogynin gets converted into 7-hydroxymitrogynin versus only about 3% of an intravenous dose. So there's 3 times more 7-hydroxy produced when you take it, ingest it orally. We also looked at metabolism of the 7-hydroxymitrogynin. I won't go into this story because it's turned out to be kind of irrelevant now. The 7-hydroxymitrogynin actually can be converted in human plasma in the blood to an even more potent opioid agonist. But this compound, we've learned, mitrogynin pseudoindoxyl, does not seem to cross the blood-brain barrier very well. And so it does not seem to have much concern for us in further analgesic or further abuse potential with this material. And so I'm going to skip going into the details of that, but did want to show you a little bit of data that shows when we give mitrogynin just by itself orally to mice, we can quantify how much of the material is in the brain and how much of the material is in the blood or the plasma. And what we can see is mitrogynin achieves a higher brain concentration than it does in the blood. And the metabolite 7-hydroxy that's generated also achieves a higher concentration in the brain than it does in the blood. So this is indicative that the molecules are getting into the brain. If they are having activity, it's definitely the central nervous system. We also gave pure 7-hydroxymitrogynin orally to these animals. And interestingly enough, when you give 7-hydroxymitrogynin by itself, you see what's called a flip-flop. So 7-hydroxy doesn't seem to cross the blood-brain barrier as well by itself. And we have less 7-hydroxymitrogynin in the brain than we do in the plasma when it's given by itself. So all that to say, we really still don't know what's going on, if there's transporter-mediated phenomena at the blood-brain barrier that can change that distribution when you give mitrogynin versus just 7-hydroxymitrogynin, the metabolite alone. But the big question was, is there still enough 7-hydroxy being generated to have an effect? So we did some pharmacokinetic modeling, which is really one of the strengths at the University of Florida's College of Pharmacy, particularly down in Lake Nona campus, where we have the Center for Systems Pharmacology and Pharmacometrics, which really is a fancy way of saying modeling of drug effect. And what we can do is look at the concentrations that we see of mitrogynin in the animal versus the analgesic effect. And so this can give you the brain concentration versus the maximal percentage effect. And we can develop a window, which are these green dots, to show you where those concentrations need to be in order to receive those analgesic effects. So when we look at that and we look at 7-hydroxymitrogynin concentration derived from mitrogynin, we see that it never really achieves a high enough level. Maximum concentration only falls in that range at very low doses. And so as we go, we never really see enough 7-hydroxymitrogynin generated from just pure mitrogynin to account for all that analgesic effect. So the take-home message about mitrogynin, the one alkaloid, is that it's really an atypical opioid. It interacts with opioid receptors, but it also interacts with many other protein targets, and so has non-opioid pharmacology. It does have some effects, but not all effects that traditional mu-opioid agonists like morphine or heroin have. It has a very low potency with very low efficacy. I showed you a little bit of that data. I did not show you that it has less tolerance than morphine, so you can utilize it longer than you can utilize morphine before you see the absolute need to increase the dose to still get the same effect. It has less dependence than morphine. I didn't show that. It has a discriminative stimulus effect that's different from morphine, so we can train rats to discriminate and be very distinct connoisseurs of morphine. And then you can give those morphine connoisseurs some other drug and ask them, does it feel like morphine to you? And in this case, mitrogynin does not substitute for morphine. So there's less tolerance, less abuse, less dependence, liability than other opioid analgesics. And we don't think that mitrogynin itself has abuse potential or addiction potential. And again, I didn't show the data, but it does reduce morphine intake in rats, which are desired characteristics of a compound around this area. The take-home message of 7-hydroxymitrogynin, though, is very different. It should be considered alkaloid or constituent with high abuse potential that may also increase intake of other opiates. It's a very selective opioid ligand. So unlike mitrogynin that interacts with serotonin, alpha adrenergic and dopaminergic receptors, 7-hydroxymitrogynin may be one of the most selective opioid compounds I've ever seen in my life. And I've been doing opioid chemistry for over 25 years now. It's pretty amazing, exquisitely selective. And again, the extent that mitrogynin is converted to 7-hydroxymitrogynin remains to really be understood in terms of the pharmacological ramifications and what that means. And I realize we're approaching the hour, but I wanted to kind of bring this back to the full Kratom product experience. And so here we can compare pain relief. This is the same thing we looked at before, dose versus percent pain relief. If we look at morphine, either injected intraperitoneally or given orally, the dose is about the same and the effect is about the same for either route of administration. We have brewed tea and then we freeze dried that tea, so it's like astronaut kratom now, and we deliver that orally to these same mice. And what you see is we can go up to 1, 2, and 4 grams per kilogram of weight. These are whopping doses, but we never really get a full efficacy like you see with morphine in terms of analgesia with the entire lyophilized kratom tea. So it seems to have a ceiling effect, and that we still don't really understand as well. It does have long-lasting analgesia, so in these mice, we can give these doses and see analgesic effects for up to about 70 minutes. That is significant. And we know that that analgesia is mediated through opioid receptors, and I won't go into the science behind this just due to the time. The other thing that's interesting from a drug abuse or drug seeking perspective is this data. And so lyophilized kratom tea or LKT lacks a condition place preference. So condition place preference is a model that we use of drug seeking behavior where we take essentially a shoebox, divide it into 3 sections. One section in the middle is a very small vestibule where you put the animal initially, and then you open up 2 doors that allows them access to 2 sides that are both the same size. The only difference is the floor may have a different texture, and one may have black and white horizontal stripes and the other may have black and white vertical stripes. We let them get to the point where they have no preference of which side they want to be in that box when it's just free association. And then we will compartmentalize them to one side of that apparatus, give them drug, and we'll do that for several days. And then we give them a day period of time to wash out. We put them back in that vestibule, open up both doors, and see if they spend their time in the box side where they got the drug, or if they spend their time in the opposite side, or if they just don't have a preference. And so what you can see with morphine, and you can think of the 0 line here as that vestibular area where they just hang out. And then the big increase here in red is the side that morphine was paired with. And so these animals will definitely go in and try and seek that experience again in that environment. What we see with the lyophilized kratom tea at large doses is they have a slight preference. It's not statistically significant from pre-treating them. So we don't think there's a drug-seeking behavior associated with lyophilized kratom tea at these doses. We also don't see any sedative properties, which is what is here. We're looking at a latency to fall. I won't go into detail because of time. We also don't see depression of respiration until, and I won't go into the purple bars there because we're looking at knockout mice and other pharmacological treatments. But just at the blue line, you see a little bit of respiratory depression in the initial 20 minutes, but it's nowhere near to what morphine is. And then that writes itself back to what essentially saline looks like. And so we don't see the severe respiratory depression that you see with these. We can also do rapid detoxification in mice that we habituate or get addicted to morphine. And we do that with naloxone or Narcan precipitated withdrawal. So we get them addicted to morphine, then we can give them a rapid injection to make them go through withdrawal. And essentially what we're showing here is that we can do 4 days or 7 days of morphine sulfate and then switch over to kratom tea. And in these assays, we see much less withdrawal sign. So jumping frequency becomes one of the major things that's obvious to see in these animals when they go into rapid detox. And we pretty much wipe that out with kratom tea treatment. Teeth chattering is another one. And And in some of the dosing regimens, we didn't see a difference in teeth chattering, but in others, we see where we could almost completely abolish that withdrawal syndrome. So we do have hope. This drug and alcohol dependence paper that was published in November of last year really underscores the potential for therapeutic treatment of opioid withdrawal with kratom. So I mentioned it was a symphony. There's all these alkaloids. How many are actually getting exposed into the blood? We actually believe that there's only 5 molecules that are being exposed into the blood when you take the full product. 7-hydroxymitrogynin is there, but it is not there in the actual tea. So it's below the limit of quantification in the tea that we process. We see mitrogynin, of course, that's the major alkaloid. We see speciosialatin, which is the second highest percentage of alkaloid. Speciogynin and painanthine we don't see in the full material because they actually are metabolized and cleared out of the system within the first hour. But interestingly enough, and I don't have time to go into all the details, but I'll just tell you speciosialatin is also a fairly potent mu-opioid agonist. So another one of the compounds that's present in the plant material is having potent analgesic effects. And then a couple of the other molecules, painanthine and speciogynin that I mentioned, actually interact very strongly with serotonin receptors. And what we've learned is these may be the ones that are responsible for the mood elevation effects or the positive effects and anti-anxiety effects that individuals report with kratom. And so they both interact with 5-HT1A receptors at a high level and 5-HT2B receptors at a high level. If anybody knows 5-HT2B receptors in detail, this is an anti-drug target. You don't want to have compounds activating this target because it causes cardiovascular complications if it activates that target. So we've gone in and really dug down into understanding what's the interaction at the target here. And these compounds are actually blocking that target. So we're not concerned about the cardiovascular implications of those. And then lastly, these molecules that are activating serotonin receptors also have analgesic properties that are non-opioid mediated. Excuse me. So the serotonin receptor itself can actually relieve pain. And so this really opens up a new door for us in terms of looking at novel ways or novel chemicals to potentially target pain and chronic pain treatment that would be non-opioid mediated. And I won't go into this whole explanation here, but essentially what we're looking at is being able to use a mu-opioid antagonist to block the effect of these drugs. And the only one that it can block the effect of is fentanyl, which is a mu-opioid agonist. The rest of these are serotonin agonists, and you still get full pain-killing properties from those molecules. So non-opioid, very promising, could be very interesting to see where we go. So I do still talk about this as 2 faces of kratom or kratom, but there's been this long history of safe use in Southeast Asia, but there's been a range of adverse events in the Western world. The compounds are definitely structurally different and pharmacologically different than the traditional opiates that we think of. And then the products in the USA are really not the same as what we see traditionally utilized over in Southeast Asia. And so there's still lots of gaps around the science in this plant. There's absolutely huge gaps in the clinical studies around this plant. And then the possible solution, or at least a possible tool in addressing the opioid epidemic, could actually come from nature, which in my mind is a perfect balance of nature, because nature gave us the opium poppy, and that's caused all this problem, and maybe nature has a solution, and this may be part of it. So lots of people do the work. There's a lot more work here that I didn't present, obviously, but it's really a huge team effort. All these individuals are at the University of Florida. except for two individuals that we've worked with at Scott Hemby at High Point University and Jonathan Katz at the National Institute on Drug Abuse. But the rest of them faculty here at the University of Florida, and we have an incredible team that can study this plant and the potential of it from essentially seed to consumer. And so we have the most comprehensive program to really look at this and really see if there's legs to it, so to speak. So we are in the process of developing plans in clinical trials. We are working with the National Institute on Drug Abuse in purified mitrogynin, the major alkaloid, and that is scheduled to be ready for clinical trials in sometime in 2022 as a pure chemical entity. And then we're working on to get a standardized Kratom product into clinical trials so we can understand the safety and the efficacy of that, and also look at potential toxicological problems that might be associated. So with that, I'm sorry I went way over time, but I felt like it was important to really cover many of the issues.
Thank you, sir, for all a very comprehensive talk. A lot of stuff I'm sure most of us are unaware of, but it points out how study of these natural products really might have very major medical consequences. And in that sense, this is extremely interesting as well for other reasons. So I don't know if we have time for questions, do we?
If there are any questions, sure.
Okay, it's open.
There we go.
Can you hear me?
Yes.
Okay. So thanks for a great presentation. Tell me about the financing of your research and interest by pharmaceutical firms in what you're doing and the implications.
Yeah, so I'm happy. We have two large grants from the National Institute on Drug Abuse. One is a cooperative agreement with the National Institute on Drug Abuse, where we actually are working with scientists at the National Institute on Drug Abuse that are helping to direct some of the scientific studies that we're performing. The other one is a more traditional funding mechanism offered by the NIH, sort of the gold standard, called an R01 award. And within those two awards, both were awarded for 5-year periods. It's been a total of, I believe, over $12.5 million to study this. The National Institute on Drug Abuse's director, Dr. Nora Volkow, is personally highly interested in the potential here for treatment of opioid abuse. We are in an interesting position in terms of pharmacological or pharmaceutical companies. One is this is a natural product, so it cannot be patented. And so there's really no commercial interest available there. However, if you have a formulation or a unique opportunity to deliver this drug in a unique way, those can be patented and then selectively utilized for single chemical entities as what would be a drug in the pharmacy. The FDA also has a new track. It's not that new anymore. It's been around for a little while, called botanical drug approval. And so we are in the process of working both tracks and trying to look at getting a botanical drug evaluated through clinical trials. And there's tremendous amounts of companies, as you might imagine, in the nutraceutical dietary supplement space that would be interested in product. We also have a farming initiative going on now, where there's a farm in South Florida that has already placed an order for 340,000 trees that will be going in the ground in the spring of next year, with the eye on the hope that there's going to be a commercial entity of a product coming down the road. Even more interestingly, and I don't want to take up too much time, but we found two different chemotypes of this plant, just like we see with cannabis. So you have medical marijuana, and then you have industrial hemp. We see a similar pattern in this tree where we have trees that are producing large amounts of mitrogynin or absolutely no mitrogynin. And individuals that have donated those trees to us claim that they're still getting all the mood elevating properties and their pain relief without having what we think is supposed to be the main active alkaloid. And so if the FDA and the DEA continue to push on the main alkaloid and that 7-hydroxy, which are the two that they're very focused on putting into Schedule 1 if they schedule the plant, this other cultivar essentially doesn't have those. And so the trees that are going in the ground are a mixture. That 340,000 trees is really a mixture of both of these chemotypes to really see which one may work out in the end of the day.
Thank you, sir.
Rod, do you have a question?
I think we're okay. Once again, we're highly appreciative of your efforts. I'll throw out two names that may ring a bell or not. One is Saul Snyder, and
Oh, yeah.
one is Bridget Kiefel.
Oh, yeah.
I could have brushed with both of them in various ways in the past, and so it's kind of interesting to see it come down to what you described today. Thank you, sir.
Yeah, thank you, and thanks for those memories. Saul Snyder is sort of one of the superstars in the field, and so is Bridget. She's tremendous, and the contributions that they've made have really changed the way we approach research. So thank you for that, and thanks again for the opportunity to be with you all.
Thank you for being with us, Chris. Stay well, everyone.
Bye-bye.
Yes, be well.